at the Marine Science Institute. However, my background is biomathematics. It's mostly math applicable to biology. And because I was interested in, I also took some bioinformatics courses. So when I came to the Marine Science Institute and was supposed to work with microbes, don't ask me for the names, I don't know them. For me, they're microbe 1, 2, 3, and so on. I got convinced they're pretty important. Microbes are not only in our belly helping us digest the food, they are also in the ocean. So if you take a glass of water, there are millions of microbes just in that glass. So, okay, they're important, but they're in the water, right? What have we to do with them? Well, actually, on average, every second breath you take contains the oxygen produced by the guys in the ocean. As they produce oxygen, they also take up the carbon from the atmosphere. And in fact, they take about half of the carbon that gets taken up from the atmosphere is done by those guys. So we should understand how they're working, how they're functioning, who is interacting with whom. And what I really like is networks. So I thought, okay, I can do that project because there are already network construction tools out there. The data is already out there. I just put them in. And what I was imagining is that each microbe we find in the ecosystem is one node in our network. And every time there is an ecological interaction between those two microbes, we connect them by an edge. Okay, so what I imagined the network to look like is like that: sparse, scale-free, and a small world. Sparse means, well, you don't have a lot of edges, right? Given the potential of edges you could have with all those microbes. Scale-free means mathematically the degree distribution follows a power law. What I find really cool about that characteristic is no matter how big your network is, if you have a small network or a huge one, you will always find a few nodes that are highly connected. But the vast majority of nodes will only have a few links. And small world means no matter which dot or which node you pick in that network, you will only have a few edges connecting to each other dot. So when we use those networks to analyze the community, how can we actually use them? We could look for really important species, so-called keystone species. However, the definitions are not standardized, so it really depends what you define as important. You could think of, okay, if a node in your network is highly connected, it has a lot of edges, it must be important, right? Probably. But what about the guy here? I don't know how to, no. That one here only has 2 edges. However, it's acting as a bridge, connecting 2 communities with each other. So it also seems to be pretty important, at least for the network. And speaking of modules, well, a module, it's a group of highly connected nodes. They are more connected to each other than to the rest. So finding these modules in your network is then it's the right time to bring back the biology and try to find out, okay, why are they more connected to each other? But I hope I convinced you why using networks to analyze microbes is a pretty cool idea. And when I started, I thought, okay, like I said, I have the data, I have the network construction tools, I put my abundance data in the tools, get the network, start analyzing, cool. But our network didn't look like I expected it to be. Our network was pretty dense. Our network was more like a hairball. And so the first thing I had to do is finding a way to get from that to here. And one approach I want to talk to you about today, I call them the bee mosquito approach, but that's like informal, and I tell you why. Imagine I go to the garden of my parents who live in a village, and I count the number of bees and I count the number of mosquitoes every week for a whole year. In summer, they're quite abundant. In winter, it gets a bit colder, right? They go down. But if you put those numbers into your network construction tools, association, correlation, you get a pretty neat correlation. Based on the mathematical model, those guys will have a high correlation score. However, correlation does not imply causation. So there seems to be something else going on, like a third factor maybe. Because in summer, when the sun rises, you have more daylight, more temperature, there are more flowers available for the bees to feed on, and also more animals out there for the mosquitoes to feed on. So the correlation is quite strong, right? But it's more indirect. We call that the effect of indirect dependency. They both correlate to a third factor, but not really to each other. And the same idea I want to use to improve the networks, because the networks we actually get from the network construction tools, they are not interaction networks, they are association networks. And we need to keep in mind that an association could mean a true ecological interaction, or it could be the same or different environmental preference. And our solution is a program that tries to find those associations that are environmentally driven and not true. And it does that by incorporating a different type of nodes, which are environmental. Here, for example, the sun. That idea is based on work done by people in Belgium. So they take an environmental triplet. Environmental triplet are 3 nodes. All of them are connected to each other. You have 2 microbes and 1 environment. And once you have that, you then apply a strategy. And that's the new thing about my method. The first one, sign pattern, also developed by the same group, only looks at the association score. The idea is what I just explained with the bee and mosquitoes. If they're all there at the same time, maybe you should remove them. But that's really strict. So I think you should combine the method with others. Overlap, that's specifically for temporal data. Interaction information goes back to the abundance data and calculates a score. And there's a 4th method that also goes back to the abundance data. I would really love to tell you more about it because I'm just thrilled. I don't have the time, so please visit me at my poster. And here are some results. So that's the network. And I should maybe mention that network is based on 10 years monthly sampling at 16S or 18S rDNA. And what we got are 40,000 edges, and my program only removed 5,000. That's 14%, but remember the associations we determined whether or not to remove them, they have to be in the triplet. So within those triplet edges, we removed about 17%, and then you need to keep in mind when we don't have the environmental factor responsible for the environmental preference, well, then we also don't detect them. I have another data set that it's not temporal but spatial. There we removed about 30% of the edges we looked at. And okay, that method has been applied in a strategy of filtering by Anders Graberot, he's in Oslo. And all that was the stuff we removed. He did a really, really strict filtering because he was interested in the core association of the marine microbes. And that's what he was left with. And the color represents the seasonal preference. And there seems to be 2 modules. Speaking of modules, now let's bring back in the biology. It seems to be that here we have a winter community and here a summer community, which aligns with findings where you don't use networks, you just look at the abundances, and indeed microbes have seasonal preferences, not all, some love the winter, some the summer, and we can see that in the network. I used the same data, but I didn't do the filtering as strict, and what I believe is if you have 10 years of data, why do you have one network representing all of it? So I'm really keen, and that's what I will do in the next months, hopefully just weeks. I would like to kick out or use the static but bring back in my time series that I have. So I would like to bring back the dynamic. And that's a preliminary result, but I'm really thrilled to show you. That's the same data, not as highly filtered. And every time the temperature goes down, so we have winter, that community down here comes up. Every time we approach summer, the community here comes up. And as I said, that's 10 years, and it seems to replicate each time. And yeah, you are the first guys I'm showing that, so congratulations. I hope you're as thrilled as I. And when you like to talk about network visualization or improving networks, or if you have a standard data set that I could test my program on, please come and speak to me. I will be at the poster session today at number 39, and on Tuesday it's M35, and I want to quickly acknowledge the collaborators of that work, which is my project supervisor, Ramiro Logares, the people in Belgium, Caroline Faust, Jeroen Reis, and Gibbs Lima Mendes, and Anders Grabarot in Oslo. And I got some help with the visualization to quickly get it done with the people in France, Erwin Delang and Marco Boudin. And if you like to see some networks popping up now and then on your Twitter, please follow me. Thanks.
Very nice talk. Time for some questions. Yes, please.
Okay, something with cycle actions, right? The network is based on 16S, 18S. And the way I did that was I built it, or the one in Oslo did because it takes ages, so I took the network he already built it. He built the network based on the whole 10 years. Because you need quite some data to get good correlations. And then I went back to the time series, and the time series says which microbe is there at which time point. So I went back to the network and just grabbed a sub-network based on who is there. So that's how I created that network visualization. However, I think you get better results if you use metabolic data, and that's why I'm in France and working with that. And indeed, you seem to see a cycle going on each winter, each summer. I would like to confirm that with the metabolic data, just to see if it's really like a cycle and not just like a one and off thing. I'm not sure if that...
So about these cycle trends, within these sub-time series that you pulled out, they look stationary or...
Yes. Yeah. Each month, it's 2 hours away from Barcelona. Each month, people go out there, they collect a bunch of water, put them through a filter, do all the sequencing. But it's just once a month, yeah.
Yes.
Is your model a kind of embedding algorithm?
Well, I had to use existing models because my boss said, well, you have a lot of other things to do, doing a new model, you don't have the time. That network is based on ELSA, which is, after transforming the data, they have a score, but mathematically, I think it's more like Pearson correlation. Pearson, yeah.
All right. Thank you, Ina.
Thanks.
Please,